Hello, today we'll be talking about metabolite biosynthesis and metabolic bioengineering. I hope I make this easy to explain and not too convoluted. We'll be talking about primary metabolites, secondary metabolites, how these compounds vary chemically, what is metabolic engineering, and how are secondary metabolites biosynthesized, because primary metabolite and secondary metabolite biosynthesis varies pretty, uh, pretty vastly. Okay, so primary metabolites are essential compounds, your carbohydrates, your lipids, and as such, these biosynthesis pathways are very, very simple, or at least should be. And here you have a very complex biosynthesis pathway, and as you can see, these highlighted molecules are your 13 primary metabolites. And as such, your organism likes to keep a flux of these compounds, as they are the starting point of most biosynthetic pathways. So obviously, as you know, your carbon sources, your pentose, your glucose, every biosynthetic pathway originates from these. But also, as you can see here, stuff like oxaloacetate can give you aspartate family molecules and whatnot. So in terms of your bacteria life cycle, these are primarily biosynthesized in your lag-log growth phase. Why? Because they're essential to growth. You need them. Some examples are obviously your carbon sources, your metabolic intermediates like acetyl-CoA, which is valuable in the Krebs cycle, and succinyl-CoA, which you need for energy production, like your GTPs and your ATPs. So central metabolic pathways such as glycolysis or photosynthesis or the TCA cycle keep these compounds in a constant state of flux. And you'll see this word flux be thrown around many times because it's very vital in metabolic engineering to understand. So what is metabolic engineering / systems biotechnology? Either or works. So since these pathways, these molecules are less complex, they're monogenic, they can be modified very easily. So you know how you have product A and then product B, and then you have a singular enzyme that codes for the conversion of product A to B. You can modify this enzyme very easily since it's monogenic. So we can remove feedback inhibition. This is very, very common where this product D, if it is overproduced, it can be toxic or inhibitory to the cell. So the cell has a naturally evolved mechanism to stop this by inhibiting enzyme activity from negative feedback, right? From this D binding to enzyme, and then it's inhibited. It can't make any more product. You can fix this by site-directed mutagenesis. You can just edit the code for this enzyme, maybe give it a missense, maybe changed an amino acid in its sequence that still gives it its functional property of turning A into B, but just not its response to D. Or you can use artificial selection using analogs. So you get a cell culture, you uh, artificially add an analog of D, and then over time, that cell will naturally evolve to be resistant to this negative feedback inhibition since D is needed. And that's why we used an analog, because if we just added the D, um, if we just added product D, it wouldn't do anything because the cell is happy. It's like, oh, we have enough product, I guess. But since it's an analog and not actual product D, it needs to evolve to actually make this product. Uh, Sorry for the yap, but that's basically the gist of it. We can remove competing pathways um, via gene knockout, so via recombination events, right? We can knock out this gene that makes acetate, and we can knock out this gene that makes PHB, which both take away the flux from pyruvate, which is a precursor for B-lactate, which is our metabolite of choice that we want. And now you might say, hey, Aaron, why don't we just inhibit the transversion of pyruvate to acetyl-CoA, right? If we do this, we don't need to knock these two out. We can only make one change and have the same effect. Well, again, as I said earlier, acetyl-CoA is a primary metabolite. The cell needs this for growth. So yes, the flux that you will get from beta-lactate will increase drastically, but also cell growth will be greatly reduced. And so the cost just outweighs the benefit. We can remove post-transcriptional control, and here you can see the lac operon, which is a classic example where the lactase, which is an isoform, allolactase, which is an isoform of lactase, actually promotes the transcription of these enzymes, which then makes more lactase. You can also have the reverse effect of this, where the product inhibits transcription of these enzymes, right? So feedback inhibition is you're stopping the enzyme from working, and post-transcriptional control is you're stopping the actual enzyme from being transcribed, from being expressed, which we don't want. We want the majority of our cell life cycle to be spent making our product. We do not want this transcriptional control. We do not want, as soon as we actually start making the product, the whole process is stopped. That's very annoying. We don't like that. We can also, you know, increase resistance to ethanol in E. coli, which is a polygenic trait, right? Because you're engineering what's it called? Oh, I'm so bad at biology. The resistance to ethanol, which is in the cell wall, like the cell membrane. Yes, cell membrane. Oh my gosh, I'm so sorry. Yeah, which is a polygenic trait because you have all these little intermolecular membranes, intermembrane proteins, which help its resistance to ethanol. You can artificially select for that by just growing E. coli in ethanol and seeing which has the most growth, and then just choosing for that over and over and over again. Holy stutter. Okay. Anyways, here's an example of what we can do. So in this example, we want phenylalanine to be produced, but then here we have some feedback inhibition where Phe, this enzyme is inhibited. So we can remove that again by side-directed mutagenesis. Knock out competing pathways. So gene knockout of these pre-phenate aminotransferase. Knock out this gene that codes for this enzyme. Something that I didn't mention in the previous slide is carbon source commitment. Since we are making so much of this product, this pathway is under more stress. So phosphoenolpyruvate, that's a tongue twister, and erythrose 4-phosphate, we can increase the presence of these carbon sources. So this pathway is more used. Something that I also didn't mention is you can efflux this product, which reduces cell inhibition and cell toxicity, because this is a natural defense mechanism of this cell. I'm sure high concentrations of phenylalanine isn't really too healthy for cell growth. So if we can find a way to efflux this from the cell into the culture medium or wherever it is you have, that will be beneficial to growth again. So that was all our primary metabolites. Now, secondary metabolites are the non-essential compounds that a cell can be auxotrophic for. What do I mean by auxotrophic? Is we knock out a gene that codes for the enzyme needed to make that product, right? And if this was a primary metabolite, the cell can't be auxotrophic for that because it needs that metabolite. But since it's a secondary metabolite, it doesn't really need it. It can be auxotrophic for it. It doesn't need to produce it naturally. So they typically have more complex structures and synthesis pathways, which result in them being a polygenic product. And we'll see this in the next slide. So here we see penicillium is an example of secondary metabolite, right? The same way as germicidin is like a inhibitor of sporulation. So naturally, these secondary metabolites would only be made during the stationary phase, when your cell runs out of natural resources and it needs to scrounge, it needs to fight other cells, other bacteria for those limiting resources. So there are many different types of secondary metabolites. We'll go over some of them. We have your alkaloids, which are a type of heterocyclic compound, which contain at least one nitrogen atom. And depending on the position of this nitrogen atom, it changes the chemical properties of your alkaloids, which can also be polynuclear, as morphine is, which, as you know, is a powerful um, pain relief. And it can be mononuclear, like pyridine, which is used as a solvent in the industry. You can also have organosulfur compounds, which again are heterocyclic, which just contain a sulfur atom in their structure. And you can have allele sulfides. These are present in stuff like garlic and onion, and gives it its pungent and antibacterial properties. And you can also have glucosinolates, which are in cruciferous vegetables, which makes them bitter and kind of a deterrent from eating them. But you should eat them anyway. And you also have polyphenolics, which are made of many phenolics, which are just a benzene ring and hydroxyl group. And these are used for antioxidant or anti-inflammatory purposes. You can find them in fruits and seeds. Cool. So what is the genetic basis of these secondary metabolites, and how did we actually find this? Well, not we, but people much smarter than me, than we. So gene libraries were made in order to find the functional genes for secondary metabolite production. What does this mean? It just means the genome of this mouse was split into many different sequences, and these were cloned into many different plasmids in order to make a genomic library. And then what we did here is we have a mutant strain which doesn't show the wild type phenotype, and we transform one by one these plasmids into this mouse and seeing if it restores the wild type phenotype. So this is what we did. These genetic clusters were found to be within the plasmid that restored wild type secondary metabolite function. This is a pretty tedious process, but it helps. And then from this process, we were able to find clusters like this, which actually encode for secondary metabolites, secondary metabolite structure. And it looks very complex, but you don't really need to know everything on this. The main aspects are core peptides are coded for, and the chain is extended. So here you have the core biosynthetic gene, which makes actually the carbon structure with enzymes like malonyl CoA, which help add the carbons to the amino acid. And then relaxed specificity tailoring enzymes modify this chain. I'll mention why I use relaxed specificity in a bit. But yeah, and then secretion and resistance features are also encoded for by this cluster. Yeah, pretty complex, pretty cool that they were able to find this and pinpoint which gene does what. So, combinatorial biosynthesis is actually based off of this discovery that secondary metabolites were controlled by this cluster. It's because we can modify this cluster, right? So enzyme activities are altered at the genetic level in order to produce novel secondary metabolites. You can do this by site-directed mutagenesis to produce antibiotic variants. So if you mix and match different tailoring enzymes, right, different tailoring enzyme sequences, you can do this because they have a relaxed specificity. They don't pick and choose which um, primary carbon chain you have to have in order to make a new antibiotic. And then I have this article here, which I'll link in the description, which goes over all of these um, different tailoring enzymes and stuff. Yeah, that's basically it. Thank you. Um, sorry for stuttering. I'll get better at speaking soon. If you have any questions, ask me. Uh, but yeah, metabolites are pretty cool. I think you should learn them or something. Okay, bye bye.